So a major interest that we have in our lab is understanding immunological memory to the infections that we have gotten as children or even as adults, and the vaccines that we get as children. Immunological memory is the ability of the immune system to respond much faster upon re-exposure to the same pathogen, and that's the basis of vaccination. So if you get, as a child, you get a polio vaccine or a measles or mumps vaccination, then you're protected from that disease for many, many years, sometimes for life. And the reason for that is because you've got memory, you've got T cells and B cells that recognize that pathogen and will protect you immediately upon re-exposure to that pathogen. So our lab has been interested in understanding how long-term immune memory or immunologic memory is maintained after either infection or vaccination. So what we'll cover in my presentation today are 4 things. First, I'll talk to you about immunological memory. Then I'll talk to you about, in particular, the properties of memory T cells, what makes memory T cells do what they do, which is protect us from infections and rapidly respond. So something about the properties of memory T cells. And then I will switch to what for us was a very exciting line of research, which is what happens if there's a chronic infection. So start with immunological memory, then talk about functional memory T cells you get after an acute infection or vaccination, and then tell you about what happens if there is a chronic persistent antigenic stimulus as you have during chronic infections or in the conditions of a cancer with the constant stimulation by tumor antigen. And then I'll tell you about an inhibitory receptor that actually results in dysfunction of these T cells during a chronic infection or in a tumor setting. So it's like a brake that comes on and keeps them from being functional. And then I'll talk to you about how you can restore function in these cells by releasing that brake. And then I'll end by the clinical implications in treatment of cancer of using the PD-1 directed immunotherapy. So what I'll share with you today are some studies that we have done on understanding T cell memory, in particular cytotoxic CD8 T cell memory. T cells are composed of 2 major subsets: the CD4 T cells, which provide help for antibody responses predominantly, and then you have CD8 T cells. These are T cells that recognize infected cells and can kill these infected cells. A CD8 T cell is the best mechanism that we have, or the best soldiers that we have to eliminate either virally infected cells or tumor cells. So this is a very potent way of eliminating your infected cells. These CD8 T cells can actually, they will attach to the infected cell or to the tumor cell. They do this because the T cell receptor will recognize peptides. They can either be tumor antigen peptides, or they can be peptides coming from the virus. For example, if the cell is infected with HIV, there will be peptides from the HIV virus which are expressed, presented by MHC class I molecules on the cell surface. So here you've got a piece of the viral peptide sticking on the surface of the infected cell. You've got a T cell which recognizes that particular peptide presented by the MHC class I molecule. That interaction will result in that T cell then killing that infected cell. And so these CD8 T cells are referred to as cytotoxic CD8 T cells or CD8 T cells. So when I talk now for the next 15 minutes or so, I will use these terms interchangeably. I might say CTL or I might say CD8 T cell, but it's the same thing, a cell that can kill and eliminate a virally infected cell. So we use this mouse model of acute infection to study the properties of memory CD8 T cells. And what we found was that memory CD8 T cells basically can live for the life of the mouse after vaccination or after the infection is cleared. And we have then extended these studies in humans and shown that people who got the smallpox vaccine even 70, 80 years ago still have memory CD8 T cells that recognize the smallpox virus. So long-term T cell memory is maintained not only in mice but also in humans and can last for literally 50 years or longer. So you get so then the question was, how are these T cells maintained? And work that we did along with other people showed that the T cells are maintained two ways. One, they are actually intrinsically very long-lived. They can live for many, many years. And most of the time, they're just very quiescent. They're not dividing. They're just sitting as quiescent cells. By quiescent, I mean they're not replicating or dividing. They're sitting as a cell that's in a G0, G1 phase. But they also undergo what we and others have termed a homeostatic proliferation. That is, they have a stem cell-like property where they can self-renew. So every few months in a mouse and every few years in a human, there's a capacity of these cells to self-renew. They will undergo another division to maintain their numbers. So if there's a slow attrition or death of these cells, there's also a slow birth of these cells that maintains numbers at fairly constant levels for many, many years. So this is one intrinsic property of a memory T cell. It can self-renew itself, and that's how it's maintained for such a long period. The other very interesting property of memory T cells is that when they now see the infecting pathogen to which they can recognize, they make a very rapid effector response. And we are dealing here with cytotoxic CD8 T cells. So even though this memory cell looks like a very quiescent cell sitting without any replication, as soon as it sees the invading pathogen to which it can recognize, it very rapidly elaborates effector function. And what I mean by effector function is that it can now very rapidly kill that cell. So a memory cell has two properties which are very different from that of a naive cell, that is one that has not gotten this program of being a memory cell. One, it's much longer lived intrinsically. It has the capability of this homeostatic proliferation to maintain its numbers, and it has the ability to elaborate effector function very rapidly. And that's the basis, that's the cellular basis of memory by CD8 T cells. So while we were doing these for many years, my lab has been interested in this question for almost 30 years now. I was at UCLA where we first started these studies, and then I moved to Emory University in 1995. And for the last 20 years, we've been continuing these fundamental studies on T cell memory, both in animal models and in humans. And obviously, these have a lot of implications in terms of optimizing and developing different vaccines. So one of the challenges in research is how do you pick a topic? How do you pick a model? So I think here the key thing is you should ask an important question. You should ask a question that a lot of people are interested in. You should ask a question that has broad implications. And that's, I think we were lucky. I was lucky that when I started my lab, I asked a question that perhaps was a good question to ask. And the question I asked was, how do you maintain immunological memory to an infection you got or a vaccine you got 20 years ago or 30 years ago or even 50 years ago? How is this memory maintained? So I think the question was a very good one. But then you have but the next stage is how do you study that question? And the advice I would give is pick a model where you can really study the question, where you can really dissect it, you can understand it in some detail. So we again, I think, made a good choice. We picked a mouse model. Sometimes people don't like what you learn from mice, and not everything you learn there extrapolates into humans. But still, I think if you ask the right question, do it well, those discoveries will hold as you move up towards human studies. Okay? So we picked a mouse model, and why did we pick that particular mouse model? We picked it because that was a natural, this virus that we used, which was LCMV, is a natural pathogen of mice. You can find wild mice in the nature which are infected with this. Of course, we were not working with those mice. We were working with mice in the lab that we infected with LCMV. But the reason I emphasize that it's a mouse virus is pathogens evolve with its own host. So if you're studying a mouse virus in a mouse, you're really looking at the interaction between a virus and its natural host. And this is much better than sticking a human virus that doesn't replicate in mice or replicates poorly. So if you want to discover fundamentals of immune responses, do it with a natural host, natural pathogen, and a virus that will replicate efficiently and give you all the things that you need to study in terms of viral infections. So I think it's picking the model, and then you ask the right questions. In this case, it was, I shouldn't ask questions. I should say you should then study what you want to look at in as much detail as you can. And again, the mouse model allows you to do this. In this particular one, reagents were available for analyzing the T cells. Reagents were available to look at what the virus was contributing. So it was a well-defined model for studying immune responses to viral infection. And then it's interesting how science moves forward. It depends on how technology is being developed. So our lab was never developing new technology. We are immunologists, more I would say biologists. We are biologists asking biological questions. We were not creating any tools, any new reagents. But others were. The science moves forward because many people do many great things. So we were greatly helped in many different things. So I mentioned to you that we did this gene expression profiling. Of course, that we didn't develop that, but those tools became available. Work done other places in terms of looking at the appropriate single-cell assays developed to look at expression of different cytokines. So the techniques and reagents that developed, one of the most important reagents that was developed actually was work that John Altman, who's a faculty member at the Emory Vaccine Center, did when he was at Stanford University as a postdoc. And that was the development of these MHC class I tetramers. These are reagents that you can use to identify specifically T cells reacting against different viruses. Okay? So this development of the tetramer reagent greatly helped in the analysis that we did. So my advice there is that as you ask the right question, pick the right model, and be aware of all the wonderful technology that's developing around you, because you have to apply then the state-of-the-art techniques. No matter who's developed it, you don't need to be an expert in it. You just bring the technique into your lab, or you collaborate with those people and move it forward. What happens if instead of there being an acute infection or a good vaccine that gives you this long-lived memory responses, what will happen if you have a persistent infection? That is, you have a chronic infection, like you have in HIV, hepatitis B virus infection, or hepatitis C virus infection, or you would have in many tumors, because the tumor antigen is there continuously. So in an acute infection setting where the T cell is stimulated for a short period, it gets all the right cues and gets all the right buttons are pushed, so you generate these highly functional memory cells. What would happen if that antigen does not disappear and continues to stimulate the T cell? So before we made the discovery that T cells are functionally exhausted, it was known, work that actually we had done and many other people had done, is that if there is a chronic infection and you look at, you try to measure a T cell or even sometimes a B cell, but certainly when you try to measure a T cell response against that particular chronic infection, you couldn't detect those cells by functional assays. Because that were the only assays that were available. And the assumption was that either the T cells are not there or they were never generated. So the fact that there was a decreased immune response against a chronic pathogen was known. In fact, we had published a paper on this many years back in the 1980s. Okay? But the basis for that non-responsiveness was not known. And most people had assumed that the T cells were not there, that they were deleted. In fact, that was a favorite hypothesis, that you delete antigen-reactive T cells or self-reactive T cells in the thymus, and those you have really this clonal deletion, and you don't have the T cells. Or the virus did something to inhibit the generation of this T cell response. So immune suppression by the virus to inhibit the response, or the deletion of these cells were the two major mechanisms for not being able to control a chronic infection, and that, of course, also extended to the tumor setting. So we then again modeled these studies in the mouse model that we used to study the good memory cell generation. And then we got a very interesting result. We found that when there is a chronic antigenic stimulation, the T cells actually get functionally exhausted.
And that was a paradigm shift, because if the T cells are there, that means you can try and fix them. If they're dead, there's nothing you can do. If they were not generated, there's not a whole lot that you can do. Okay? So that was a paradigm shift in terms of showing that the T cells are physically present, but they're dysfunctional. So the T cells are physically present, but now they are no longer able to kill the infected target cell. They are no longer able to secrete cytokines which have antiviral activity. So both the cytokine ability to secrete cytokines which have antiviral effect, that's lost, as well as the ability to kill the cells is lost. And also their ability to proliferate and expand greatly after antigen stimulation is lost. So we defined, ours was the first group to define this T cell exhaustion. This was a paper we published in 1998. Actually, this is a paper that was published from Emory University. Alan Zayak, who was a postdoc in the lab, made this discovery that T cells are present, but they are dysfunctional and are functionally exhausted. Yeah, so one of the joys, I should say, of science is that if you kind of make an interesting discovery and then see how it moves forward. So for us, the T cell exhaustion part that Alan Zayak, there was excitement in the lab that we had made this discovery and allowing the possibility of maybe fixing these cells. And then when Dan Barber did what I think were truly seminal experiments with the blockade, there was huge excitement in the lab. Dan was very excited. Everyone in the lab was excited that we could do something so simple. Because the experiment was very, very simple. We had a chronically infected mouse, and all Dan did was he injected some antibody into the mouse. A very unsophisticated experiment that anybody could do, just a single injection. And then the result really was quite striking. After we reported the finding that T cells become functionally exhausted during chronic stimulation in this mouse model, several other investigators at other places and also at Emory University showed that the same finding of T cell exhaustion occurs in human infections. So it was then shown that people who are infected with HIV have HIV-specific CD8 T cells in their blood that you can easily detect, or even in their lymph nodes when some lymph node biopsies were done. Okay? But they are not able to kill the target cell, so they become dysfunctional. Other investigators showed the same thing in chronic hepatitis C virus infection, hepatitis B virus infection. So it became very quickly established that human chronic viral infections like HIV, HCV, HBV, which have very high levels of antigen, high levels of virus, and persist for the life of the individual unless they get treated with appropriate drugs, okay, you get this T cell dysfunction. Also, people at around the same time who were working in cancer models, tumor models in mice, showed the same thing. They showed that there is T cell dysfunction in the T cells which infiltrate the tumor. These cells are referred to as tumor-infiltrating lymphocytes. TILs is the abbreviation that's used. So it was shown that TILs also have this T cell dysfunction because of the chronic stimulation by the tumor antigens. And then oncologists who were working with human cancer patients found the same thing. So this phenomenon of T cell exhaustion, which was first described in this mouse model of that we use, is a virus called lymphocytic choriomeningitis virus. You don't need to worry about what the name is or what it does. It can infect humans. It's really of not much significance there, but it's a wonderful mouse model to study many different aspects of the immune system. So these studies which came out of this LCMV mouse model of T cell exhaustion were quickly extended into human cancer T cells and in human chronic viral infections. The next question was, what is the mechanism of this T cell exhaustion? Why are these T cells not able to kill the target cells? Why are they not able to do what the good memory cells do? So the contrast here was with the highly functional memory cells with the dysfunctional cells under chronic infection. So what we then decided to do was that we said, let us look at the gene expression profiling of a highly functional memory cell and compare it to that of a highly dysfunctional cell. And the system that we used allowed us to take T cells of the same specificity. And what I mean by that is it's a T cell which has a T cell receptor
which sees the same peptide from the virus that we're studying, but one is under the conditions of an acute infection and the cells are highly functional. Another is under conditions of a chronic infection where the cells are dysfunctional. So we're not comparing apples and oranges, but we're comparing different flavors of the same fruit. Okay? And so we looked at the 20,000 plus genes which are expressed by these cells, and we found that there were some striking differences in the gene expression profiling of the exhausted cells compared to the functional cells. And there are many, many differences, but what we were struck with, and that was the thing that really was the most differentially expressed between these two cells, was the expression of inhibitory receptors. So there are inhibitory receptors that a cell can express, and these inhibitory receptors actually inhibit T cell receptor signaling. T cell receptor is what recognizes the tumor cell or the infected cell, and its signaling through the T cell receptor is needed for the cell to then kill the target cell. So these are inhibitory receptors that come up and are regulating that T cell receptor. And the most striking receptor that we identified in terms of T cell exhaustion was a molecule called PD-1. PD-1 stands for programmed cell death 1. This was a gene that was cloned by Tasuku Honjo in Japan in the early 1990s. The gene had been around for a long time, but its function in terms of chronic infections, its function in terms of T cell exhaustion was not known. So our studies identified that this molecule was expressed at very high levels on these dysfunctional or exhausted CD8 T cells. But then what really clinched the deal here was in a collaborative study with Gordon Freeman, who's a scientist at Dana-Farber Cancer Institute at Harvard Medical School. Gordon Freeman had antibodies that could bind the inhibitory receptor. And the way the inhibitory receptor works is that this PD-1 will bind to a ligand that's either on the tumor cell or on the infected cell. This ligand is known as PD-L1, L standing for ligand. So it's a molecule that will bind the inhibitory receptor. Okay. So what Gordon had made was antibodies that would either bind the inhibitory receptor or the ligand and prevent the interaction between these two. So if you prevent interaction of the PD-1, which is on the T cell, the PD-L1, which is on your target cell, if that interaction is blocked, then the inhibition does not happen. And the results we got were really quite striking. We showed that if you take a chronically infected mouse and you infuse this mouse, you inject into these mice antibody either against the inhibitory receptor or against the ligand, disrupting that inhibitory interaction, suddenly now you restore function back in the cells. The cells begin to expand because they have the TCR stimulation. They are able to kill the virally infected cells, secrete antiviral cytokines, and most importantly, bring about a reduction of the viral load. So this was the first demonstration that this blockade of this PD-1 pathway in vivo under condition of a chronic infection will restore function back into the T cells and result in the control of the viral infection. So this finding had a huge impact. This work was done actually by a graduate student at Emory University. Dan Barber was the first author of this paper, and this paper attracted a lot of attention. This was published in 2006. Immediately there was a flurry of papers that came, and some of those we were involved in collaborating with our friends, showing that PD-1 also is involved in inhibiting HIV-specific T cells. This was a study we did with Bruce Walker at Harvard. And then in studies that we did at Emory with Arash Grakui, who's a faculty member at the Emory Vaccine Center, Arash showed that the same thing happens in HCV-infected people. People who are chronically infected with hepatitis C virus do have CD8 T cells that recognize HCV, but they have very high levels of PD-1 on their cell surface, and that inhibits the function of those T cells. Others showed the same thing with hepatitis B and so on. So basically it was very quickly established that T cell exhaustion, as I told you, happens in many chronic infections, but now it is also shown that a major regulator of the T cell exhaustion is this inhibitory receptor PD-1. And that blockade of this interaction results in function in those cells. So think of it as a brake that's coming on the T cells. So the T cell receptor, you're pressing the accelerator because there's antigen stimulation, but the brake is on. So basically nothing happens. And releasing PD-1 is releasing a brake on the T cells. And people who were working in tumor systems also made the similar observation that PD-1 is a major target on the TILs, these tumor-infiltrating lymphocytes that get into the tumor but cannot kill the tumor, have very high levels of PD-1. Then there was a lot of interest in extending these in vivo experiments from mice into non-human primates and so on. So actually we did a study with Rama Mara, who's also a faculty member at the Emory Vaccine Center, and Rama showed that if you give an infusion of this blocking antibody to monkeys that were infected with SIV, SIV stands for simian immunodeficiency virus. It's a cousin of HIV, and basically it's the HIV virus of monkeys. And what Rama and his group showed is that if you block PD-1, PD-L1 interaction in non-human primates, you get an increase in the function of the SIV-specific T cells and a reduction of the virus in these monkeys. In a collaboration we did with a group in Ohio, with Chris Walker, we showed the same thing in the HCV model of chimpanzees. That's really the only model for HCV that can be done. And then Chris showed the same thing happening in HCV-infected chimpanzees. And as I mentioned, a lot of people working in tumor models showed the same thing. And then what has been quite amazing to see is the very, very, very rapid advances that have happened in terms of clinical trials of PD-1 in cancer patients. This, of course, is not our work. We don't work in cancer models. We actually never even worked in a mouse cancer model. Okay? But the real benefit of PD-1, and the progress has been quite remarkable, has happened in the last 3 or 4 years. And these studies have been led by many major pharmaceutical industries, but 3 in particular, Merck, Bristol Myers Squibb, and Genentech. These 3 big pharmaceutical companies have developed anti-PD-1 antibodies and also anti-PD-L1 antibodies. I told you both, in effect, do the same thing. They block this inhibitory interaction and release the brake on the T cell. And the results have been quite promising. PD-1 blockade has been shown to be effective in both phase 1 and phase 2 clinical trials for multiple different cancers, in particular for melanoma and for lung cancer. In particular, lung cancer really had hardly any if therapy was there in terms of chemotherapy. All of that was really not that effective. But but the PD-1 has given promising results in 15 to 20% of the treated people. That's not a very large number, but when you go from basically no survival, 0% survival, to 15 to 20%, that's really quite a dramatic change in the treatment of cancer. And within 3 or 4 years of these human clinical trials starting, both Merck and BMS have a licensed drug. So PD-1 was licensed last year for melanoma treatment, actually a year and a half back, for melanoma, and last year also for lung. So this has been quite remarkable to see, and I give this as an example for how science should be done. When we first started down this path many years ago, 25, 30 years ago, our interest was very simple. We asked, how do you maintain T cell memory long term? We were not thinking of cancer, we were not thinking of immune therapy. That was completely out of our radar. But these fundamental studies on understanding T cell memory led to T cell exhaustion. The T cell exhaustion led to the linking of PD-1 with T cell exhaustion, and then a potential therapy for the rescue of those exhausted cells. And that has very quickly really work done, and the credit for this goes to the oncologists who did these clinical trials at different places, these companies who moved things very forward very quickly. But essentially you now have drugs which are effective in treating melanoma and also in treating lung cancer. In conclusion, I would like to say that this has been a fantastic journey for us and the wonderful team of people that I've had in my lab. That's really, for me, that's one of the best things, to see these people do great work and they're going to move on and set up their own labs and flourish on their own. And in terms of where we are now, in terms of understanding T-cell exhaustion and understanding how to restore function back, and in terms of clinic, I think we still have a ways to go because we would want to be able to get much better response rates in the cancer patients. We want to be sure that there is durability of the treatment that they're getting. So here would require greater understanding of the T-cell exhaustion mechanisms. I would say maybe beyond PD-1, what can you do beyond PD-1 to take it to even to the next stage? Combination therapies with either cytokines or other inhibitory receptors or with therapeutic vaccination are possibilities. But there are other ways also. There is work that we and others are doing looking at the epigenetic landscape of exhausted cells, and could we change basically the insides. The PD-1 actually is working at the outside, but inside can we change the chromatin of the cell? Can we change the epigenetic landscape of the cell? So these are not easy to do, but there are ways of doing this. There are enzymes that methylate, for example, the DNA that regulate gene expression. So we have some preliminary data that modulating some of these methyltransferases will change the function of the cells. So there's very many exciting and challenging things that can be done to what I would call changing the insides of the cell. So the goal would be release the brake and change the insides of the cell. The next stage now is that why doesn't everyone respond? Why is it only the rates are for melanoma? Rates actually are quite high. Melanoma responses are as high as 40% response rate in late stage melanoma. But then you ask, what about the other 50%? Why are they not responding? I think this is for us the next challenge. Are there other inhibitory receptors that need to be identified and the right combination tried? Or do you need to provide some other kinds of stimulus? Do you need to combine the PD-1 blockade with a therapeutic vaccine, with a vaccine against the cancer antigen? So this is a very active area of research. Many companies, many groups, and we're also exploring these things. And how can you take it from being 30%, 40% out to 80% or 90%? That's a very, very high bar, but I think that's what the goal will be for us and for all the other people working in this area. The other question is, and for this we don't have the answer yet, PD-1 has only gone into cancer patients in the last few years. And as I mentioned to you, in lung cancer, the response rates are between 20% in that range, for melanoma about 40% range. But the question is, so instead of their survival being 3 months now, they're still alive, 2 years or 3 years. But the question is, how long will be the durability of this response? Will the cancer again come back? If it comes back, what the thing will be? So there are two issues that need to be addressed. How long will be the durability of this effectiveness? And how can we get a response in the patients who don't get the response? So I think this science here is very exciting. I would encourage any medical student who looks at this to go into the area of cancer immunology, fascinating area where you can do basic science and also see applications in the clinic very quickly. And again, I think the main message I want to give you is do fundamental discovery work, and that will result in something useful.